Okay, so today I'm going to talk about a couple of drugs that I've been involved with. I'm going to talk about the discovery of amlodipine. Amlodipine was the fourth best-selling drug in the world and achieved sales of $5.5 billion. And then I'm going to talk to you about the discovery of Viagra, and I will tell you the true story. So amlodipine is a calcium antagonist. So what is a calcium antagonist? We have calcium channels throughout our body. We have calcium channels on various tissues. We have calcium channels on our arteries. And if we block voltage-sensitive calcium channels, that inhibits excitation, contraction, and coupling. And that relaxes vascular smooth muscle. And if we relax vascular smooth muscle, we lower blood pressure. So a calcium antagonist will lower blood pressure. But we can also relax the coronary arteries. And if we relax the coronary arteries, that increases blood flow to the heart, and that is very good for treatment of angina. And therefore, with one mechanism, calcium antagonist, we can develop a drug that can treat hypertension, high blood pressure, and also to treat angina. But as I said, calcium channels are present throughout the body, and they're also present in the heart. And if we block the calcium channels in the heart, we depress the heart, and reduce the force and the rate of contraction. So if we want a calcium antagonist, we want one that is selective for vascular smooth muscle and doesn't interact with cardiac muscle. Now when we looked in the literature, there were various calcium antagonists already reported. This is a molecule called nifedipine. It has a very short duration of action, and it has to be given 3 or 4 times a day, which is very inconvenient. But there were a lot of patents, there were 90 patents around this structure, so very difficult to do something that was new. So we looked at nifedipine as a starting point, and we thought that might be very difficult. There's also a compound called glutazione, which is a calcium antagonist. But as you can see, the structure is completely different, and when we looked at the pharmacology, we found it wasn't a calcium antagonist. And then there's another structure called verapamil, but for the medicinal chemist, it's not a very interesting structure, and the ratio of activity on the periphery versus the heart is low. So it's a very difficult compound to administer. So we come back to nifedipine. But what's wrong with it? It has a short duration of action, and that's because it's rapidly metabolized, and I'll come on to that in a minute. It's not very soluble in water. It's insoluble. Therefore, when you take it by mouth, you get different degrees of absorption. And also, so you've got a compound here which is erratically absorbed and has a short duration of action. Now, we want a compound that can be given once a day. We don't want a compound that can be given 3 or 4 times a day. So when we looked at our starting points, excuse me, the only one we came back to was nifedipine. So we've got to make the compound more soluble, more metabolically stable, better bioavailability, and once daily duration. So that is the challenge. We first have to understand what is the problem. Well, when nifedipine passes through the liver, there is an enzyme in the liver called cytochrome P450, which you may have come across. Cytochrome P450 removes the hydrogen from here, and oxidizes the dihydropyridine to the pyridine. And this is biologically inactive. So what we want to do is slow down the rate of that oxidation process. Now the oxidation process is carried out by an enzyme that has an ion, a heme moiety at the center. And the dihydropyridine has to approach the heme moiety to be oxidized. So what we thought we'd do is we put something at the 2 position here, something large that would prevent the molecule approaching the ion and would stop the oxidation. We also thought that if we had a hydrogen bond here, that would stabilize the molecule, and if we had something here that was basic, that would give us solubility. So don't forget, we don't know the structure of the calcium channel that we're trying to interact with. All of this is imagination. We have to imagine what the molecule might look like because we don't know the target. We don't know the structure of the target. So what we're going to do is we're going to make compounds that have substituents here, which, as I said, will give us better solubility and a reduced rate of oxidation. The next thing you have to do if you're doing a drug discovery program is you have to have a screening sequence which is relevant to the clinical situation. And our first screen is the rat aorta. We can track the rat aorta, and then we look at the ability of the compound to relax it. and that tells us we have a calcium antagonist. We don't want activity on the heart, so our next screen is going to be heart. That tells us straight away the potency of the compound and the selectivity of the compound. We then go into the anesthetized dog, and we look at the effect on coronary and peripheral hemodynamics. What does it do on blood pressure? What does it do on coronary flow? What does it do on heart rate? Now, because we want a compound that is 100% bioavailable and with a long half-life, we go into the dog to do pharmacokinetics. So we measure the absorption of the compound and the duration of the compound, and that tells us how it will perform. And then there isn't an animal model of angina, so we go into the hypertensive dog, which has high blood pressure, and we look at the ability of the compound to lower blood pressure and the duration of action. All of those screens are relevant to the clinical situation. If we get a compound that comes through those screens with those criteria, we will have a compound which is fit to go to the clinic. So then, when we carry out medicinal chemistry, it's very important that we understand what other people have done. We're not an island, we're part of the scientific community, and it's very important to understand what other people have done. And I remember going to the library one day and reading a paper called Arsenine Forschungsmittel, a journal, and I noticed this calcium antagonist called itafanone, and it had a structure here which I thought would fit perfectly on the dihydropyridine. So straight away I went to John Stubbs, who was a chemist, and said, John, why don't we put that moiety on the dihydropyridine? And John said, that's a good idea. I've just read this paper by Kellogg, and I know how to do the chemistry. If I'd gone to John 6 months earlier, he wouldn't have read the paper. If I'd gone 6 months later, he would have forgotten the paper. So Pasteur's dictum is chance favours the prepared mind. If you have an idea, take it to your colleagues and develop it. Don't sit on it. It's really important you engage your colleagues. So that's what we decided to do. So first of all, we looked at the potential for introducing substituents here in the 2 position. We put in an amide with a basic center. This was potent, and by that I mean it's nanomolar. It's selective tenfold. It was soluble, but the pharmacokinetics were not very good. So we then put on the structure that I showed on the previous slide. Here we've put on oxygen, 2 carbons and a dimethyl amino. This is again potent and selective, and the half-life is 8 hours in the dog. The half-life of nifedipine, which is where we started, is half an hour. So we've gone from half an hour to 8 hours with rational chemistry. But it was not very well tolerated. The dogs didn't like the compound. They were very unhappy with the compound. But rather than stop the chemistry, we tried to understand what was the problem in the dog. And what was happening in the dog was that one of these methyl groups was being removed to give you NH-methyl here. That had a longer duration of action. We were getting accumulation and we were getting toxicity. So we thought, well, why don't we nominate the NH-methyl, which we did, and we got the same problem. So finally, we decided to nominate UK48340 amlodipine as our candidate, because the only metabolism carried out here is oxidation to the pyridine ring, and the pyridine is completely inactive. So that was our candidate. I've shown you the design principle, and I've shown you the screens that we used. Now, if you look at this structure here, it's not very informative. But if we look at the 3D structure, you can see here's the dihydropyridine ring. Here's the other aromatic ring. They're orthogonal. They're at right angles to each other. Ester groups are normally unstable, and you normally don't have an ester group in a drug. But because the lone pair of electrons on this nitrogen is delocalized into the esters, they are stable. They're not hydrolyzed. And because of that delocalization, this nitrogen here is not basic. And the distance between the oxygen and the hydrogen is about 2 angstroms, so there probably is a hydrogen bond that we speculated about. So that's our compound. So here now is a very important slide. We're looking now at rat portal vein. And we're contracting the rat portal vein, and we're looking at the ability of the compound to relax. So here's the control. You can see tension is maintained over the test period. We give nifedipine, we get a very rapid onset of action, very short duration and a very rapid offset. And that's exactly what you see in the clinic. Blood pressure drops very rapidly, very short duration, and then returns. And when your blood pressure drops rapidly, you get a tachycardia, an increase in heart rate, you get flushing, you get a headache, and it's not very pleasant. And now we're going to evaluate amlodipine in the same screen. And as you can see, at 1 hour, the compound is rather disappointing. But the person running the screen, Simon Tickner, said, I'm going to run the screen at 3 hours and see what happens. And Simon let the compound equilibrate with the tissue for 3 hours, and you can see here we have a good response, a good relaxation, and here you can see we have a very slow offset, which is exactly what we want. So when you're doing an experiment, you have to follow the protocol, but also modify the protocol if you want to ask different questions. And here we wanted to ask the question about activity at 3 hours, so Simon Tickner modified the protocol, and this is what we got. Other people evaluated amlodipine, but they found it to be inactive because they evaluated it only at the 1 hour time point, when you have to evaluate it at 3 hours. Now here's some pharmacokinetics, and I won't go through it in detail, but we're changing the X group, we're changing the Y group, and we're changing the Z group. And here's amlodipine, which I've shown you before, and it has a half-life of 30 hours. So this compound is a once-daily calcium antagonist, which is 100% bioavailable. If you look at other members of the family, phenodipine and nicardipine, they have half-lives of 1 hour. Now the half-life is made up of 2 components. Clearance, that's the rate of metabolism, and here you can see the clearance of amlodipine is slower than the other 2 dihydropyridines. So as I showed you previously, we probably slowed down that metabolic process. But the other component of half-life is volume and distribution. And you can see here the volume, and this is the ability of the drug to get into tissue. So non-steroidal anti-inflammatories will stay mainly in the blood and will have a low volume of distribution. Compounds like amlodipine will get into the tissues where the receptors are, and they have a high volume of distribution. And as you can see, it's a very high volume distribution. So if you take reduced clearance, high volume, you get a 30-hour half-life. And nobody else has ever made a compound in this series with a half-life greater than 1 hour, greater than an hour. So this is a major achievement. Normally, volume distribution is related to lipophilicity, or the fatty nature of the molecule. You can see here we have a one-to-one relationship with all these other members of the series. Amlodipine is an outlier, a much higher volume of distribution than you would have expected. And why is that? Well, when the molecules equilibrate with the tissue, there's a hydrophobic interaction, which recognizes the dihydropyridine. But we believe amlodipine is protonated, so this is positively charged. In the membrane, there are phospholipid head groups which are negatively charged. So we've got 2 interactions now. We've got the hydrophobic interaction, we've got the charged interaction here. That's allowing the molecule to stay in the membrane longer and giving us a higher volume distribution. So everything we try to do is rational. If you see a property which is unusual, try and understand it, don't just ignore it. Okay, what does it mean for patients? Well, if you take nifedipine, as you can see, you get a very rapid onset and a very rapid offset, but you're very quickly into the plasma levels for side effects. People usually take more than they need to. You get into the plasma levels for side effects, you get a tachycardia, you get a flushing, not very pleasant. If you take amlodipine, it takes 7 days to build up to steady state, so the patient doesn't know that he or she is taking it, and it doesn't accumulate. So you take amlodipine, it's a very slow onset, very slow offset, you don't really know you're taking it. If you forget to take this dose of nifedipine here, you have no protection, because this dose here is worn off. With amlodipine, if you forget to take a dose, you just take another dose the next day. Now you probably know cardiovascular events, stroke, heart attack often take place early in the morning, 8 or 9 o'clock. If you take a compound like this with a short duration, you don't have any protection in the morning. If you take a compound like amlodipine with a long duration, you have protection. So the compound is very beneficial to the patient. And the other thing is, compliance is the big issue. Patients forget to take their medicine. And if you're taking it 3 or 4 times a day, you forget, you don't want to do that because it's inconvenient. If you're taking it once a day, that's much more convenient if you take it at the same time. So here we have very good effects on compliance and very limited side effects. Now, I won't go through this in detail, but what we've done is we've changed the X group, we've got oxygen, sulfur, and we've got carbon. So all we've done is we've changed one atom in the molecule, just one atom in the molecule. And as you can see, we've gone from a half-life of 30 hours down to 8. So just changing one molecule is very important. So when you're doing chemistry and discovering drugs, it's very important you do molecular changes in a very careful and considered fashion. And the reason that the half-life has gone down is that the volume distribution is different. And probably with amlodipine, this head group here is in a tight conformation, whereas with these two compounds, it probably flops out into space. So again, just a simple molecular modification can give you significant changes in properties. So this is the final slide. This may be too much chemistry, but we started with a nitro group here. We've changed it for chlorine. This is an asymmetric center, and only one of the enantiomers is inactive. We changed the methyl group to increase potency. We put in an oxygen atom here to give it a hydrogen bond. And we put in an ammonium function here, which is basic, protonated physical and intercellular. So the compound is 100% absorbed in all patients, has a 30-hour half-life, and is once daily, and it became the 4th best-selling drug in the world, with annual sales of 5.5 billion. And it's known as Norvasc, or amlodipine, which you may have heard of. Sorry. Excuse me a minute. All right, that's all I'm going to say on amlodipine. Let's move on now to Viagra. So when we take a compound to the clinic, like amlodipine, we know we're not going to get 100% response from patients. You never do. There's always patients who don't respond. So we thought to ourselves, we take amlodipine, it was very successful, right? Now, what other mechanism can we think about that might be useful in angina? How else could we treat the patients? And there were very few options. But we came across cyclic guanosine monophosphate. And cyclic guanosine monophosphate was said to relax smooth muscle, which is exactly what amlodipine does, and also to increase blood flow. So we thought to ourselves, if we could increase the tissue level of cyclic guanosine monophosphate, we would relax smooth muscle, would increase blood flow. We'd have a new treatment for angina and hypertension. Now, cyclic guanosine monophosphate is formed from guanylate cyclase, and we thought the stimulant here was a peptide. And we don't want to work with peptides because they don't make very good drugs. But we also knew that cyclic guanosine monophosphate was broken down by phosphodiesterase 5. So we thought if we inhibit the action of this enzyme, then we would increase the levels of cyclic guanosine monophosphate, would relax smooth muscle, and would increase blood flow. It's a very simple idea. Nobody else had worked in this area at the time. Where are we going to start? Well, there was only one starting point, a compound called Zaprinast. One of the very few cyclic GMP-PD inhibitors available. It was developed as an anti-allergy compound, which says it's probably got dirty pharmacology. But it did relax smooth muscle, and it did lower blood pressure. Now, it was relatively weak. It was only micromolar, and there are also other members of the family, and it inhibited those as well. So we have a weak and non-selective starting point. So what we did with, we looked at the distribution of electrons in Zaprinast and in cyclic guanosine monophosphate, and we thought that this ring here could be a mimic for the guanine ring in cyclic guanosine monophosphate. Now here's the cyclic phosphate, and there's nothing in this molecule in this part of the active site. So what we're going to do, we're going to change this structure here to make it a better mimic of the guanine, and then we're going to introduce substituents which will occupy the space of this group here. Now in 1985, remember the date, 1985, David Roberts and I wrote a research proposal saying, let's seek inhibitors of phosphodiesterase 5 for the treatment of angina and hypertension. So that's where we started. So the chemistry was taken up by Dave Brown, Nick Terrett and Andy Bell, and I won't go through it, but they made a lot of variations here. And let me just summarize in taking, this is the heterocyclic ring of Zaprinaz. What we've done is we've taken out a nitrogen, we've introduced a methyl, and we've gone from 2 micromolar to 0.3. So we've increased potency by tenfold, just by rational chemistry. So let me just remind you what I said before. Here is cyclic guanosine monophosphate with a cyclic phosphate. We've now changed the heterocycle. We're going to introduce something here and something here that will increase potency even further. So what we did, we changed the methyl group to propyl, and we've got another tenfold increase in potency. And then we introduced the sulfonamide, and now we've got a compound which is 3 nanomolar. So we've increased the potency 1,000-fold by rational chemical design from where we started. So let me show you the selectivities. As I mentioned, there are several members of the PDE family. We have about a 10-fold selectivity over 6, PDE6, but we don't hit PDE3, and PDE3 is present in the heart. And if you hit PDE3 in the heart, you'll get arrhythmias. So we don't hit PDE3, which is good. I'll come back to PDE6. And we did some pharmacology, still thinking this was a cardiovascular agent. We showed that cyclic GMP levels were raised in the coronary artery. We showed the compound was antithrombotic, and we did some pharmacokinetics. And we decided, therefore, this is a novel mechanism of action. Let's take the compound to man. So here we are. Remember, we started the research proposal in 1985. We took the compound to the clinic in 1991, and we didn't find any cardiovascular activity. We were very disappointed. It's a new mechanism of action, and people were saying, stop the project, it's not going to work. We said, no, we're going to do a 10-day study in patients at the maximum tolerated dose, and we're going to find out what happens. And if nothing happens, we'll drop the compound. So we did a study in students in Cardiff, in Wales, and we did a 10-day study, and when we asked the doctor who was running the trial, what have you seen with this drug? The answer came back, not very much. We've seen headache, we've seen muscle pain, we've seen gastrointestinal disturbance. Oh, he said, and by the way, one of the young nurses has noticed erections. Oh, so we didn't know what to do about that. Why was a compound causing erections? Was there a need for a compound of this sort? So our commercial people went out and said, yes, erectile dysfunctional impotence is a big factor in breaking up marriage and relationships, so there is a medical need. Well, the next question is, how do you measure the effect? You can't go around with a ruler. So how do you do it? So what we did, we designed a questionnaire for the patient, and the patient with erectile dysfunction always lies. We designed a questionnaire for the partner, and the partner never lies. So we were very good there. Then the question came back, how does the compound work? We don't want a compound with no mechanism. We don't want a compound that's an aphrodisiac. We need to understand how this compound is working. And again, Pasteur's dictum, just at that time, there was some evidence that if you look to the corpus cavernosum, the smooth muscle in impotent patients, the relaxation of the smooth muscle was impaired. And here we've got a drug that relaxes smooth muscle, so we can start to understand. It was also shown that if you inhibit the synthesis of nitric oxide, then that will inhibit erections. And also shown that if you give patients cyclic guanosine monophosphate, it will improve erections. So again, Pasteur's dictum, chance favours the prepared mind. Everything is coming together. In 1992, we're in the clinic. Nitric oxide was the molecule of the year. And I can show you now on the slide, why does Viagra enable an erection? Well, in the penis, there's a local release of nitric oxide, which stimulates guanylate cyclase. That forms cyclic GMP. That relaxes smooth muscle, inflows blood into the penis, and that maintains an erection. And you can see immediately, if we inhibit the enzyme, which is breaking down cyclic guanosine monophosphate, we will potentiate the effects of nitric oxide and we will enhance erections. So immediately now, we've got a lovely, simple, concise mechanism, and we understand what the drug does. Here's a little bit of biochemistry, just showing the 1,000-fold increase in potency. When we were doing the experiments in the lab, we used the PD5 from the rabbit platelet. But we then had to show that there was a PD5 in the penis, and we did that, and you can see the potency of the compounds are the same. As I said, we did show there was a PD5 in the corpus cavernosum. This is now, we're looking at rabbit corpus cavernosum, and we're looking at the effects of sildenafil, and this is cyclic guanosine monophosphate, and you can see we're getting a nice dose response increase in cyclic guanosine monophosphate, and this is exactly what we want the compound to do. Now we went into patients, and you can see here patients reporting improved erections, 10, 25, 50. You can see here we've got a nice dose response. Now, those of you who are still with me will notice that the patients who received a placebo, so they got the tablet with nothing in it, and 40% of them reported increased erections. So the placebo effect is very significant. Mm-hmm. If you have a new antidepressant, for instance, and you treat patients with the new antidepressant and you have the placebo group will get better because you're paying attention to them. So when you're doing clinical trials, when you train to do clinical trials, it's what goes on in the placebo group is important, just as important as what goes on in the treated group. And here, if you have a drug which you say is going to be good for erectile dysfunction, people imagine that it's going to work and they don't know whether they've got the placebo or not. Now, erectile dysfunction can be organic, mixed or psychogenic, and you can see here we're getting very good responses to Viagra, and the placebo response now is more respectable. Erectile dysfunction often coexists with diabetes and hypertension. You can see here we get a nice response, and the placebo group is coming down. Radical prostatectomy, the response is not very good. When the surgeon goes in and takes out your prostate, he or she often takes out the nerves around the penis. So there is less nitric oxide being released, there is less space, if you like, for Viagra to work, and that's why we don't see good response. These are the people who have broken their backs, had motor car accidents or sports injuries, and we get any number of letters from these people saying, I'm in a wheelchair, but I'm taking Viagra and I'm getting married. It completely changed my life. And so we think this is a very serious drug. We think it should be used on prescription. We don't think it's a recreational drug. And in 1998, I appeared on the Juan Suarez show in Rio, and you know Juan Suarez? And he kept pushing me and pushing me and pushing me to say that this was a recreational drug. But in the end, it isn't a recreational drug. It's a very serious drug for a serious condition. I won't go through this in any detail. You can look at any of these parameters you like. What we're doing is we're bringing a subnormal parameter up to normal. We're not taking a normal parameter and making it supernormal. Side effects we have to report, and you can see here, headache, flushing, that looks serious. But if you look at the discontinuation rate, the people who stop taking Viagra, very low, same as placebo. So really no significant side effects, no cardiovascular effects. There is a little bit of altered vision, and this is because we have a PDE6 in the eye, and inhibiting PDE6 leads to a difficulty in distinguishing red from green. But it only occurs in a few subjects, and it's completely reversible. So we started the program in 1985, and in 1998 I was lucky enough to be in New York when we launched the drug, and we launched the drug on my birthday, March the 27th. So it was nice to have been with the research proposal right from day one, and then launching the drug on my birthday, March the 27th, 1998. And as you know, Viagra's made a huge impact on social conditions. The sales, I think, became $3 billion, the most popular drug in its category in the world. Very important to say that drug discovery is multi-team. So here are the chemists, here are the biologists, and here are the clinicians. And it took us from 1985 to 1998, 13 years from idea to market. So quite a tough road. I'm just going to make some general remarks in the last couple of minutes. I made this slide in 1992, and in 1992, 2020 was a long way off. But now 2020 is very close, and what are we going to die from in 2020? Well, ischemic heart disease is still the big killer. Although it's dropping in the West, as the developing nations take on our diet, the incidence of ischemic heart disease is increasing, and it's still the #1 killer. If you look at diabetes, there are 100 million diabetics in China, so diabetes is a huge problem, and obesity in India is a huge problem. So, there's still some big challenges facing. Cerebrovascular disease or stroke. We've taken 100 compounds to the clinic for stroke, and I'm not talking about the thrombotics, the thrombolytics. We've taken 100 small molecules to the clinic for stroke, and not one has worked. And the reason is the animal model we have for stroke is just completely inappropriate and not related to the clinic. And chronic obstructive pulmonary disease was #6, it's going to be #3, and that's been driven by the millions of smoking-related deaths in China. So smoking is bad for you, and you can see the increase in the deaths in China is going to drive this up. So there are still great challenges for medicinal chemists, great challenges for physicians like yourselves. How will therapy change? I think there's still going to be a role for small molecules. You've obviously come across antibodies. They're very important, although they're expensive, but I think they'll continue. And of course, proteins are important. So I think small molecules, antibodies, and proteins. Gene therapy has proved to be very difficult. I think there's still some time off. Ribozymes and antisense and RNA interference, very difficult to deliver. I think there is a role for vaccines, but I think there's still a role for small molecules, antibodies, and to some extent proteins. And you may have heard over recent years that the pharmaceutical industry has been going through some difficult times. And I think the problem with the pharmaceutical industry is that it industrialized drug discovery. For every 10 compounds we take to the clinic, only one becomes a drug. So let's take 100 compounds to the clinic and we'll get 10 drugs. So the pharmaceutical industry is automating everything and making everything high throughput. So instead of hundreds of compounds, thousands of compounds, millions of compounds. And I think in that process, the individual contribution has been lost. So here we have, this is the pharma model. Just make the numbers, make the numbers. This is my model. I actually think it's a Brazilian, isn't it? My model is, give me the best technology. I have to have the best technology. I'm not going to win Formula 1 in a full storm. Give me the best technology. I need a strong leader here, someone with vision and someone with strength. But you win or lose races in the pits. You lose 0.1 of a second in the pits, you've lost it. So teamwork is very, very important. So technology, leadership, and teamwork. And I think drug discovery is a shared experience, not a mechanical event. And pharma has treated it as a mechanical event. And that's why I think we've seen many problems in the pharmaceutical industry. What's required for success? I think teamwork, teaching, every conversation should be a learning experience. Idea sharing, don't lock your ideas away. Information bites. I think people are too consumed now with too much information. You need to create time to think. You need to create time to look out of the window. It's very easy to spend, your day on email or on your mobile phone, but I think you've got to create time to think. And I think that's very, very important. Speed, you've got to be fast and size. When I left Pfizer, there were 10,000, after they merged with Wyeth, there were 10,000 people in research. You can't carry out innovation on a scale of 10,000. I like a Roman centurion that looks after 100 people. And that was the size of the group that I liked. So don't get into groups that are too big. Make, working groups where everybody knows each other, you know what they're doing scientifically, you know what they're feeling personally. So that's my model. And then where did all this happen? Well, this happened in Sandwich in Kent. And Sandwich, the Shrubscope, the Earl of Sandwich was playing cards, and he said to his butler, go and make me something to eat. So he took a piece of bread, a piece of ham, and another piece of bread, and that's where a sandwich comes from. So this is Sandwich in Kent. This was a port, but the sea now, the river silted up, so the sea is here, 3 miles away. So these are the original Elizabethan buildings from 1600, which are absolutely perfect, and Pfizer is just down the road here. So I had an incredible career at Pfizer. I can't imagine anything more interesting, more stimulating. I'd like to thank all of my colleagues in Pfizer and in academia that I work with, and I'd like to thank you for your kind invitation today and for your attention. Thank you.